Hi there, everyone. We are so happy to have you, whether you are joining us here today or watching later on our Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already. We will have time to answer questions at the end of the presentation. My name is Monica Joanna Almacave, and I am the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential treatments for GABA-A variants by partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trial. Today, we welcome Dr. Juliet Knowles, one of our Cure GABA-A Recommended Physicians at Stanford University. Dr. Juliet Knowles will provide valuable insights about the research program at Stanford and help us understand the genetic epilepsy program. As a distinguished researcher, Dr. Knowles leads a dynamic lab at Stanford focusing on the mechanisms underlying pediatric epilepsy. Her research group is particularly interested in understanding generalized epilepsies, monogenic epilepsies, and developmental and epileptic encephalopathies. Notably, her lab recently uncovered an unexpected contribution of activity-dependent myelination to the progression of generalized epilepsy. Currently, Dr. Knowles' lab is investigating whether similar mechanisms are at play in various forms of genetic epilepsy, including GABA-A-related epilepsy. I'm handing it off to you, Dr. Knowles. Please share with us your research and how it relates to GABA-A.
All right, thank you so much, Monica, for that kind introduction. I'm so delighted to be here. As Monica told you so eloquently, I'm really delighted to have this opportunity to talk to you about research and clinical care for pediatric genetic epilepsy at Stanford. So just to give you a roadmap of the seminar today, I'm going to start by just giving you a flavor of what my research lab does at Stanford. And, most of our work so far has focused on a mechanism called myelin plasticity in generalized genetic epilepsy. And I'll tell you a little bit about that, how myelin plasticity is a newly appreciated form of brain plasticity, but we realized that it could also play a role in epilepsy. And I'll show you some of the actual experiments and data that we obtained indicating that, in fact, this activity-dependent myelination or myelin plasticity may play a role in the progression of epilepsy. And our research indicates that myelin plasticity might not just be active in the one type of epilepsy that we studied. And we're looking at other forms of epilepsy now, particularly in epilepsy related to GABA-A loss of function. So that's the connection to the GABA-A Foundation. So I'll tell you a little bit about the new work that we're doing in a mouse model of GABA-A loss of function. And then in the second part of the talk, I'll switch gears and I'll just tell you about the clinical program that we have here and the genetic epilepsy clinic that we started a few years ago, which is growing quickly in the scope and the types of services and research that we're able to do here. Okay, but first, I'll start with kind of lab research on myelination in epilepsy. So some of you may have heard about myelin, but essentially myelin is an insulating substance that coats neurons, and neurons are the information transmitting cells of our brains. And if you've heard about myelination before, it was probably in the context of developmental myelination. So what is developmental myelination? This is the remarkable process of the initial myelination of our brain that begins in sort of the perinatal period, just around the time that we're born, and then occurs extremely rapidly over the first couple of years of life. And then in humans is a protracted process that actually extends multiple decades. And so we're not finished going through this process of developmental myelinations until the third or fourth decade of life. And in contrast to activity-dependent myelination, this is sort of a pre-programmed process that unfolds in our brains as they develop. And it occurs in a predictable pattern where the brain circuits that we need the most for sort of survival, they myelinate first so that they're functioning optimally. And then later, the brain circuits that serve more complex functions like judgment and things like that myelinate, in our 20s and 30s. So as an example of what this process looks like, this is an MRI scan of the brain from, a typical MRI from a 3-month-old. And what's highlighted in red here are the areas that would be expected to be myelinated in a 3-month-old. So you can see that most of the brain isn't myelinated at that point. And so here, for reference, is a picture of my son when he was 3 months old. And you can kind of see that the things that he's able to do sort of correspond to, where myelination has occurred. He can see an object, he can suck on a pacifier, he can sort of reach out with, sort of a rough grasp and grab this ring that he's looking at. But then, as I mentioned, over the first 2 years of life, this remarkable process of very rapid myelination happens that enables the rapid acquisition of skills. And so this is what the same, child's MRI scan would look like at 18 months. And you can see that the red area has vastly expanded here and much more myelination is present. And along with that, there is this rapid acquisition of skills between 3 months and 18 months, for example. So how does this happen? How is this possible? Well, it's possible because of these cells, oligodendrocyte precursor cells, or OPCs. These are remarkable cells that are stationed throughout our brains, throughout our lives. And they can respond to different signals and stimuli in the brain environment. For example, we now know that they can respond to the activity of neurons. So as I mentioned, neurons are kind of like the wires in the computer circuits of our brain. They are the information transmitting cells. And so what happens is that in response to stimuli during development, or as it turns out after development, these OPCs can be induced to proliferate and mature into myelinating oligodendrocytes. And then what happens is that myelinating oligodendrocytes can extend these lipid-rich processes that form spiral wrappings around these around the neuronal processes called axons, so sort of the wire. So this is like insulation on the wire of the neuron. And the net effect of this is that it enables rapid transmission of signals, much more rapid transmission of signals down this neuronal axon. So it really speeds up the efficiency of neuronal signal transmission, and at the level of sort of big collections of neurons in networks in our brain, this sort of increases coordination and synchrony within those networks. Now, an interesting observation is that even after developmental myelination is complete, so like in our 30s and 40s, there are substantial numbers of neuronal axons that remain unmyelinated. For example, some of the axons in the corpus callosum, which is a large white matter tract that connects the two sides of the brain, are unmyelinated. So this is a picture of axons in cross-section, and these are the myelin sheaths, these dark rims around the circles here. And you can see that there are also axons here that don't have that dark rim. And so, a substantial proportion of neurons remain unmyelinated. And this suggests that maybe we have the ability to myelinate more axons sort of on demand or as needed to help us, become more efficient in certain ways. So there have been now a series of really cool studies that have demonstrated what appears to be white matter change, potentially consistent with increased myelination in response to various learning and sort of training paradigms. So, for example, this was a very cool study in 2009 where they took healthy adults and they trained these adults how to juggle. And so they had never juggled before, but they began to juggle every day for 6 weeks. And they did MRI before and after. And this was a specific type of MRI that called diffusion-based MRI that enabled them to, get some idea of what was going on with white matter structure and myelination. And what they found was a change, possibly consistent with increased myelination in a region of the brain that's very important for hand-eye coordination, which would make sense, right? Because they were learning to juggle. And indeed, a number of sort of studies with similar ideas have been done in humans and animals, where the subjects participate in various training and learning paradigms, such as piano practice, tests of long-term or working memory, spatial navigation testing. And in each case, what they found was evidence of white matter structural change in a region of the brain that kind of matched the skill that they were trying to acquire. So this suggests that the areas that are highly myelinated in our brains, called white matter, can sort of respond to experience, and therefore the activity of neurons that underlies that experience in our brains. And this idea was formally tested by another scientist here at Stanford, or two scientists, Erin Gibson and Michelle Mange, both of whom are my colleagues. And what they did is they took mice that were genetically engineered to have neurons that will become activated in response to light. And so they activated a specific motor circuit in these mice that prompts them to walk. And they repeatedly activated the neurons in that circuit. And they looked to see, if you activate the neurons in that circuit, does that actually change what's going on with OPCs, oligodendrocytes, and myelin? And I don't have time to go through all the results of this paper, but suffice it to say that what they found was that there was indeed a circuit and network specific increase in the number of proliferating OPCs and oligodendrocytes and myelination, simply in response to repeated neuronal stimulation. And indeed, other people from around the world have now demonstrated in various ways that in the healthy brain, this neuronal activity-induced myelination is helpful. It's an important part of our brain's sort of adaptation to different stimuli. And in fact, this myelin plasticity is required for multiple forms of learning and cognition. Now, I'm somebody who thinks about epilepsy, so I'm a pediatric epileptologist. And when I learned about this research, I wondered whether it might also play an important role in diseases that are defined not by sort of helpful patterns of brain activity, but by pathological patterns of brain activity that don't serve us as well, such as seizures in epilepsy, for example. And as many of you will no doubt know, seizures in epilepsy are detectable in an EEG, such as the one shown here. And in support of the idea that changes in myelin and white matter structure have something to do with the pathogenesis of epilepsy, we know from multiple studies that have used diffusion imaging that white matter is structurally abnormal, sort of in regions of the brain that are affected by seizures. So we specifically wondered whether seizures, kind of like, the learning paradigms, whether seizures themselves might induce new myelination or alter the pattern of myelination in those seizure networks. And if that happened, how would that affect the brain after that? Might this be a part of the disease process that we just hadn't recognized previously? So to begin to study that, we have been looking at absence seizures and myelin plasticity. So some of you might know already what absence seizures are, but these are a generalized seizure type, meaning that they appear in both sides of the brain at once and occur in multiple forms of generalized epilepsy, including all of those listed here and including GABA receptor beta 3 subunit loss of function. And these seizures are outwardly manifest by brief but very frequent behavioral arrests or staring spells. And along with those staring spells, there's usually impairment of consciousness. And so you can imagine that this can be very disruptive for a child who's trying to, make their way through in the world and learn at school and things like that. And when these spells are captured on EEG, they have a generalized, so bilateral spike wave discharge correlate, such as this EEG shown here. These types of seizures, absence seizures, are commonly encountered in children with epilepsy. And the first line medication for them is typically ethosuximide. Now, we know some about where these seizures come from in the brain. So they originate as abnormal patterns of activity between a deep brain structure called the thalamus and the overlying structure, which is the cortex that's connected to it. And the structure that connects these two things is the internal capsule. And I mentioned that the seizures, appear in both sides of the brain. And that's because after the seizures start, they rapidly propagate across this major white matter tract, the corpus callosum, that connects the two sides of the brain. And fortunately, we do have animal models that allow us to better understand what's happening with absence seizures. So one example is this model, SCN8A mutant mice, which we used for many of the studies that I'll show you. So these mice have a loss of function mutation in this gene, SCN8A, that encodes a voltage-gated sodium channel that's very important for sort of controlling neuronal activity. And so these mice, like children with absence seizures, have these bilateral seizures. So you can see here we've recorded from both sides of the brain in the somatosensory areas. And on this EEG from this mouse, you can see the background is relatively normal here leading up to it. And then there's a spontaneous absence seizure with this spike wave discharge pattern. And I think you can also appreciate that it starts in both sides of the brain at once. Another feature that these mice have in common with children that have poorly controlled forms of generalized epilepsy is a progressive course. So the seizures start around postnatal day 21 in these mice and then rapidly increase in their hourly frequency until they're having 30 to 40 seizures per hour by the time they're 45 days old. And in thinking about this progressive course, we wondered, what mechanisms of brain plasticity allow that to happen, because that's really key, understanding that is key to preventing this process in children. And we reasoned that absence seizures might induce activity-dependent myelination in the seizure network. And that if that were to occur, this could actually sort of cement in that seizure pattern and contribute to seizure progression. So we tested that idea by looking at the myelin-forming cells of the brain, and we looked within the seizure network. Here, these are pictures from the corpus callosum, that big white matter tract that connects the two sides of the brain. And what we found is that mice with frequent seizures have increased numbers of proliferating OPCs and oligodendrocytes compared to mice without seizures. And this difference doesn't occur until after the seizures already start. We also, looked to see whether this was resulting in changes in actual myelination. And so these, again, are pictures of the corpus callosum, the large white matter tract that connects the two sides of the brain. And these are axons sort of in cross section. So you can picture, a wire with some insulation on it. And then if you cut it down the middle and sort of looked at it in cross section, that's what we're looking at here. So these white circles are the axons and the dark rims around them are the myelin sheaths. And we noticed that some of these myelin sheaths in the mice with seizures appeared to be thicker than what we saw in the mice without seizures. And we quantified this with something called the G ratio, which is a standard metric in the field for looking at myelin sheath thickness. And it's defined as the diameter of the axon over the diameter of the axon plus its sheath. And so what we found is that mice with seizures had decreased G ratios, and this means that they have thicker myelin sheaths. We also found an increase in the percentage of axons that are myelinated in these mice. And then we wanted to get a much bigger view, like how much of the brain is this happening in? How broad is this phenomenon? So we used advanced MRI imaging in collaboration with Dr. Jennifer McNabb, who is an MRI physicist here at Stanford. And we conducted a survey of white matter tracts throughout the brain of these mice to see how widespread these differences were. And so here we looked at the corpus callosum from the very front to the very back. And what you can see here in this yellow area is that the mice with seizures throughout the entire anterior, front half of the corpus callosum have decreased G ratios. So their myelination is altered throughout this entire anterior region of the corpus callosum. And we then went on to sort of do the same thing in multiple white matter tracts throughout the brain, to sort of survey, like, what is the extent of this? And essentially what we found is that seizure-associated myelination is widespread throughout the brain, but it's happening in very specific regions of the seizure network, particularly the white matter tracts that connect the two sides of the brain. And this actually makes a lot of sense because these are generalized seizures that appear in both sides of the brain. And so what this seems to suggest, or what this sort of hinted to us, is that maybe this myelination that is happening in these tracts that connect the two sides of the brain is enabling these seizures to occur in that progressive pattern. So we then shifted gears once we got these findings to investigate, what is myelin plasticity doing in epilepsy? And I won't go through all the technical details, so I'm happy to chat more after if you have questions about it. But to answer that question, what is myelin plasticity doing in epilepsy? We generated the same mouse model, SCN8A mutant, but we genetically engineered them so that we could specifically block activity-dependent myelination at specific time points. And so we basically started the myelin blockade after they had finished their developmental myelination, but just before seizures started. And what we found is that the mice in red, which have the seizure-causing gene and normal myelin plasticity, those mice have this dramatic increase in seizures over time, this progressive course, sort of seizures causing more seizures. But these mice in blue have the same seizure-causing gene, but they no longer have the ability to have the myelination response to seizures. And what we found is that those mice had many fewer seizures over time. And this was really intriguing because this was the first demonstration that myelin plasticity can actually promote epilepsy progression, this phenomenon of epilepsy sort of tending to get worse over time. So I'm a clinician scientist, and of course, I'm always sort of thinking about what does this mean for patients and is this clinically relevant? So I wondered if we could give a drug to block myelin plasticity after seizures have already started, and if that would be helpful. And it turns out that there is a class of drugs that we know about that can block myelination, and these are the histone deacetylase inhibitors. And basically what those drugs do is they prevent these changes in gene expression that need to happen for OPCs to become oligodendrocytes. And in fact, there is one type of histone deacetylase inhibitor called trichostatin A, or TSA, that had already been shown to block activity-dependent myelination in prior work. So we decided to see if we could block myelin plasticity with a drug and if that would help. And so we took these mice, SCN8A mutant mice, and we let them have increasing uncontrolled seizures for 1 week, kind of similar to what might happen in the real world where, when somebody comes to you for help with epilepsy, they've already been having seizures for some time. And at that point, after 7 days of seizures, we then started the trichostatin A, the HDAC inhibitor, or a vehicle control. And we treated them every day for 2.5 weeks, and we monitored what happened with their seizures over time. And what we found is that just prior to starting the treatment, mice that got vehicle or the drug were already having seizures in equal numbers. But after 2.5 weeks of treatment with the drug, the mice that received the drug had many fewer seizures than the mice that received vehicle. So similar to what we found when we genetically blocked myelin plasticity, when we blocked myelin plasticity with a drug, this also prevented seizure progression. So to kind of wrap up what I've told you about the research program that my lab's been working on, over the last decade or so, we've come to appreciate that there's this new form of brain plasticity, myelin plasticity, and that this is helpful for learning. However, our studies and our data indicate that myelin plasticity can also play a role in diseases like epilepsy. So we specifically found that absent seizures can induce abnormally increased myelination within the seizure network, and that abnormal myelin plasticity in turn then serves to further cement in that very same pattern of seizures, and the seizures become more frequent. And this was, the first demonstration of myelin plasticity playing any role in a disease process. So whereas myelin plasticity is adaptive in the healthy brain, we termed this particular mechanism that we observed in the mice with epilepsy maladaptive myelination. And some of the next steps that we're working on now, sort of building on that work that I just showed you, is to understand how this occurs. So we're using techniques like transcriptomics to sort of piece apart, what specific mechanisms are happening in the OPCs at the start of this maladaptive myelination. Because if we can understand that, then we may be able to target it, with either repurposed or new drugs. We want to better understand whether this process is reversible or how we might help to reverse the process. We want to understand also whether maladaptive myelination sort of negatively impacts helpful brain functions like learning. And again, maybe we can sort of reverse any sort of negative or unhelpful brain changes that happened to sort of reinstate normal cognitive ability. And finally, a very important question is whether maladaptive myelination occurs in other forms of epilepsy. So all the data I showed you was just from one mouse model, the SCN8A mutant model that involves one gene. And this is important because, as many of you probably know, epilepsy is really a collection of many different diseases that just have one thing in common, and that is the predisposition for seizures. And so, we really wanted to know, if this mechanism of maladaptive myelination is broadly applicable across multiple forms of genetic epilepsy. And for a number of reasons, we decided to next look at GABA-A related epilepsy, specifically beta-3 subunit loss of function. So this is probably review for most of the people on this call today, but GABA is a neurotransmitter that is very important in the brain for sort of quieting down, excessive brain activity. So there's a fine balance that our brains maintain of excitation and inhibition of these information transmitting cells that I told you about, the neurons. And if that sort of homeostasis is disrupted, you can wind up with seizures. So if there's sort of runaway excitation of neurons and not enough inhibition, that is, a set of circumstances that can lead to seizures. And so these neurons in pink are GABA expressing inhibitory interneurons. And so these neurons release GABA onto the excitatory neurons and sort of quiet them down in sort of a finely tuned coordination of activity. And GABA receptors that are expressed on neurons that respond to the neurotransmitter and lead to sort of quieting down of the neurons have this this classical structure where there are 5 subunits. You can see they're shown in different colors here that are named with these Greek letters, alpha, beta, gamma, for example. And there is some sort of mixing and matching of subunits in these different receptors. But this particular combo shown here is a frequent combination in our brains that's, has been well studied. So 2 beta receptors, 2 alpha receptors, and a gamma, or I should say subunits, 2 beta subunits, 2 alpha subunits, and a gamma subunit. And you can see that the beta 3 subunit is one of these commonly expressed subunits. So what happens then is GABA binds to this receptor, and that causes it to open. And then an ion called chloride enters through this channel when it's open. And that is what serves to sort of decrease the level of excitation and activity of neurons. So, as I mentioned, if this system is not working properly, if there's not enough inhibition, you can wind up with runaway excitation and seizures. And so GABA-A, mutations in GABA-A that sort of disrupt its function are a well-known aspect of genetic epilepsy, or a well-known cause of genetic epilepsy. And the most commonly affected subunits are alpha 1, beta 2, beta 3, and various gamma subunits, including gamma 2. And changes in these genes can lead to a number of different epilepsy subtypes: generalized epilepsy, febrile seizures, and then another type of epilepsy called developmental and epileptic encephalopathies. And of these different types, this is probably the most severe version of epilepsy because it involves sort of pervasive disruption of brain activity that disrupts sort of global aspects of brain development and cognitive function. And so for a variety of reasons, we got interested in this type of epilepsy and understanding whether some of the things that we've studied in my lab with myelin plasticity also apply here. We're interested in these generalized epilepsies, and as a clinician who takes care of children with epilepsy, I'm particularly interested in this, most severe form of epilepsy and seeing if we can make a difference there. So in terms of genetic mutations in these subunits, loss of function variants are more common. So you could have a change in these genes that either leads to loss of function, so sort of decreased function of that gene, or gain of function, so sort of amplified functioning of that gene's protein product. And this is important to know whether it's loss of function or gain of function, because that tends to play out differently in sort of how it reflects, in a child's life. So loss of function variants, which is what we study, for example, typically, will be manifest with seizure onset a bit later than the gain of function variants. You can see febrile seizures and generalized seizures with loss of function variants. They tend to have mild to moderate intellectual disability associated with them, for example, and refractory epilepsy is less common. So given the contrasts here, you can see why, it's important to understand whether a variant that someone may have is loss of function or gain of function. So in my lab, we're now studying myelin plasticity and other important research questions in a mouse model of GABA-A beta 3 subunit loss of function. And this mouse was created by a group at Vanderbilt. And they created this mouse based on a discovery that this particular mutation, D120N, in the beta-3 subunit of GABA-A, leads to one of those severe developmental and epileptic encephalopathies. In this type, it's called Lennox-Gastaut syndrome. So this mutation leads to the severe epilepsy Lennox-Gastaut syndrome in children. And so if you actually put the same mutation in mice, they have some of the same kind of disease manifestations as the children with this variant. And so these are snapshots of EEGs from these mice at 4 months old that we recorded their EEGs in our lab. So here you can see this is an example of something called an atypical absence seizure that is characteristic of Lennox-Gastaut syndrome. It's a prolonged slow spike and wave pattern. These mice also have another type of seizures called myoclonic seizures where they have these quick jerks. And then they have another type of seizure called typical absence seizures that are similar to the atypical ones, but they're just shorter and their EEG signature is slightly different. And then finally, the mice also have less frequently these large, big shaking seizures that we sometimes see in children with this same variant, where they have, whole body shaking, what we call generalized tonic-clonic seizures. And you can see that here's the EEG, and then this is a recording of muscle. So you can see that as this seizure ramps up, then you start to get, big movements of the mouse. So we're using this mouse model, as I mentioned, to study a number of different questions. So first of all, to understand whether myelin plasticity also occurs in this mouse model and whether it has the same maladaptive role as what we saw in the SCN8A mice. And we're also using this as a platform to understand GABA-A related epilepsy and mechanisms related to generalized epilepsy and DEEs and other aspects as well. So an example of another study that we're doing with these mice is in collaboration with Juliet Knowles, who is one of the world's experts on GABA-A related epilepsy in Denmark. And we are working with a very talented medical student from Denmark, Cordelia Smith. She's going to come work in my lab for a year. And she's also being advised by Christina Høj Hansen, who is another physician, pediatric neurologist and epileptologist in Denmark. And what we're going to be working on in collaboration with Dr. Muller and Hansen is looking at genotype-phenotype correlation in GABA beta-3 subunit loss of function. So, I mentioned that we're studying that in the mouse model that I just showed you. But, it turns out that there's a range of variants in beta-3 subunits. And some are gain of function and some are loss of function. So we're going to focus specifically on the loss of function variants. And Dr. Muller, her group has already done a very detailed characterization of these children that have these variants. So how severe is their epilepsy? What kind of seizures do they have? What treatments did they receive? What other types of issues did they encounter with cognition or development? And so what we can do is take the different specific mutations in that gene and see if we can make predictions about whether different mutations lead to sort of less severe or more severe phenotypes. And the mouse model that I showed you has this D120N variant, and we know that that same gene variant is present in this cohort of people. So we'll be able to see kind of what is the range of phenotypes associated with that variant as well. So that's sort of part one of the collaboration. And then in part two, we're going to take the GABA beta-3 D120N mice that I showed you that have such frequent seizures. And we're going to use a technology called single nucleus transcriptomics to try to understand what are the cellular mechanisms that are occurring during the progression of epilepsy in these mice, and then use a bioinformatics approach to see if we can identify drugs that could reverse those processes. So we're very excited about this collaboration, and that wraps up the part of our research program that I wanted to tell you about. So now I'd like to switch gears and start talking a little bit about the clinical genetic epilepsy program that we have at Stanford. So a few years ago, several of us in the Division of Pediatric Neurology realized that we were basically facing a tidal wave of new genetic diagnoses, especially in epilepsy, and we wanted to sort of meet the moment with the best resources possible. So we started the Genetic Epilepsy Clinic a few years ago now, and the main providers in this clinic are myself, Dr. Emily Spellbrink, who's another clinician scientist, Dr. Bill Galantine. He's the current chief of our child neurology division and has really spearheaded the initiation of this clinic effort. Dr. Becky Levy also works on our team. So she is a clinician scientist who's really amazing. She has been trained in child neurology and did a separate genetics fellowship. So she's been very instrumental to our group. And then importantly, we are lucky to work with Lauren Matas, who is a genetic counselor on our team. So this is our core group in the Genetic Epilepsy Clinic. And typically, if you were to come to a visit, you would see one of the epileptologists, and then Becky and Lauren is sort of the genetics part of it. Or sometimes, if we think we have a pretty good idea of the gene causing the epilepsy, you'll see the epileptologist and the genetic counselor. And I put here the website if you want to look us up, the clinic at Stanford. We have a little web page on it. And in this slide, I just want to show kind of our broad vision. So like, what are we hoping to accomplish with this clinic? Of course, we want to provide families, of children with genetic epilepsy, diagnostic evaluation and exceptional care, and cutting edge care to try to get you, the best treatment and management possible. But our vision really extends beyond that. We are at Stanford University, so it goes beyond sort of the basic providing clinical care. We also want to do research to really better understand what is causing different forms of genetic epilepsy. How do we best treat it? How can we push the field forward and provide even better treatments? So within our program, we have basic science research done by my lab and others at Stanford. We also do translational research. So sort of, either what we call bench to bedside. So you develop a discovery in the lab and then you use that to inform better treatment of genetic epilepsy. Or sometimes it goes in the reverse, but we observe something in the clinic and then we decide to study it to better understand it in the lab. And so this is kind of the space that I live in, is this world between basic and translational research. And then clinical research as well. So we participate in natural history studies where, we sort of work with other centers to put forth a concerted effort to really understand how a genetic epilepsy plays out over time, because that, helps us not just help families know what to expect, but then we can also better design clinical trials once, things like gene-based therapies become available. We can understand, the time scale that we need to measure effects over what types of endpoints we want to measure that would be helpful and sort of appropriate for the abilities of the children, for example. And who's on the team? Well, I showed you the core people. Certainly we have physicians and a genetic counselor, but really our team here at Stanford expands well beyond that. So we have clinical and research coordinators, social work, palliative care. We also work with psychiatry and developmental pediatrics and therapists here at Stanford, understanding that epilepsy is not just about seizures. There's a whole host of other issues that children and families may be contending with, including dealing with developmental delays, behavioral or cognitive issues, sleep issues, things of that nature. So we, aim to provide multidisciplinary and holistic care to our patients as well. Oh, and I want to acknowledge that this slide is courtesy of my wonderful colleague, Becky Levy, that I showed on the previous slide. And in terms of our research program, the vision really is that we want to push forward precision medicine for genetic epilepsies. So this knowledge of, the specific genetic causes for epilepsy that's become available to us with really in the last 10 to 20 years. We want to be able to use that knowledge to provide specific targeted treatments. And, sort of an abstract way, this involves understanding the gene and being able to diagnose the genetic epilepsy, understanding the natural history. So, how does a change in a gene play out over time? What are the issues that you're likely to encounter and at what ages, for example? And then, the other part of it is that we have to figure out which treatments are best, whether it be sort of maybe drugs that we already have will work particularly well for a specific genetic etiology. And so all the way up to, developing new gene-based therapies for specific disorders, such as ASO treatments or gene therapies. And then, of course, we need to do clinical trials to test the efficacy of those therapies. So, our vision is really that we can help families at each step along this way. So sometimes people are coming to us and, we diagnose them with a new, sort of very rare form of genetic epilepsy, or sometimes we already know the gene and we already know the natural history, and we're trying to help them find the best treatment. So this is what we do. We navigate along this sort of process with our families. Great. So I want to thank you all so much for the opportunity to talk to you about this work today and your attention. I want to briefly acknowledge my colleagues at Stanford and the trainees and staff in my lab that contributed to this work, and as well as the funding support for my research. And this is a picture of my research lab at Stanford. These are the amazing people that made all of that work happen that I showed you in the first part of the talk. And then again, these are my colleagues in the genetic epilepsy clinic. So if you're interested in this or want to find out more, don't hesitate to check us out online. My lab has a website that's here. And then this is the website for the genetic epilepsy clinic. And I'm also on social media. So with that, I'll stop and love to take any questions.
I just wanted to say thank you so much for your presentation. It was so easy to follow, and I'm sure our community is going to walk away with so much more understanding of myelination, and also confirming that absent seizures do get worse over time. And so thank you for confirming that.
Yeah. And understanding that a bit better, right? Like, why does it happen? Why is the brain learning to have seizures like that? And how can we begin to interrupt that process? So.
Yes. And also I will include your links in all of our social media and the YouTube channel comment section. And of course, we're honored to have you in your lab under our Recommended Physicians tab on our Cure GABA-A website. I wanted to thank you for speaking on precision medicine and what that roadmap looks like. At Cure GABA-A, we so understand how much natural history data collection is important. And we're currently doing that digitally through a platform called Matrix and also Citizen.
Yes.
So,
yes. Big fan of Citizen. Yeah, that's great. I'm so glad that you're doing that. Because I mean, multiple labs are working on GABA-A and genetic epilepsies, and we're moving forward closer each day to better treatments. But then we're going to have a new challenge, which is going to be, how do we design clinical trials for these treatments? And the more information that we have going in about what does this, disease look like in people with different mutations, for example? How does it play out? What should we be monitoring for so that we can set ourselves up for success with these clinical trials and get to a real answer? So I'm glad that you're doing that.
Thank you. I just wanted to open the floor to any questions. Does anybody have any questions for Dr. Knowles? I see a lot of thank you for the presentation coming through. I have a question, actually. What is your projected timeline for the drug screening?
Yeah, great question. I mean, I think that within a year, we hope to have some leads. So we hope that the information that we get from our transcriptomic studies, that we will basically be comparing that, using sophisticated bioinformatics, not sort of manually, because it's a huge data set, but so using bioinformatics to compare that to published data sets of drug mechanisms. And so that will lead us to some leads. And we think that we can accomplish that in 1 year. And then we'll need to test those, to see if they do in fact help. I mean, the bioinformatics approach will give us clues, but we'll need empirical evidence as well. So, if we have really promising leads, we'll be focusing particularly on drugs that are already FDA approved and safe for use in children. And, preferably drugs that have a, favorable brain bioavailability. So like you can take it by mouth and it gets into the brain without a problem, and that have low toxicity profiles, So that would be the best case scenario. And then we'll test those drugs probably in the same mouse model, and look at endpoints like seizure burden and cognitive endpoints, and see if these lead drugs help. And then, if you found something really promising, like if, a lead drug was that was FDA approved in children, had, a really striking beneficial effect in the mice, then you could pivot pretty quickly to a clinical trial.
Augustina, I see that you asked a question. Do you want to go ahead and ask Dr. Knowles?
Yeah, I just wanted to understand, like, once you get favorable information in mouse models, do you typically test in iPSC cells of like, because we are working hard to collect those from patients with mutations, and I was wondering to what extent that would collaborate in your research.
I'm so glad you asked that question, because I didn't mention in my talk that we're actually building up the capabilities here for biobanking from patients that we see, so we can bank samples here, or we can get them from outside, like from combined brain, for example. Yes, and basically develop induced neuronal cultures from those patient cell lines, and use those as a further level of confirmation that a drug is or is not likely to work. I think there are pros and cons of different model systems, right? So it's intuitively very clinically relevant to use patient-derived cells to test efficacy, right? Because you're working with human cells and a real person's genetic background, and a variant that's actually seen in human disease. And so we can use those to try to better understand how a drug is likely to affect, for example, gene transcription, or protein expression of something like beta-3. And we can get some information about how it might affect function, for example, by looking at indicators of neuronal activity, or sometimes actually then we can cultivate networks of neurons in a dish, and you can measure various metrics of network activity in a dish. In terms of measuring things like seizures, you kind of need a whole a whole intact brain, you know, to look at things like that, or cognition. So that's why we like to use sort of this in vitro cell culture model in tandem with an in vivo model that lets us look at things like seizure and, you know, any signs of toxicity, things like that, that you really need a living organism to assess.
And to just clarify for some of our community members who are new, in vivo means in
I'm glad
vitro
you
in
brought
addition.
that up, though. Yeah,
Yeah.
I don't want to bring unnecessary jargon in. Yeah, sorry. So in vivo is when we talk about, you know, in an organism, like in a mouse, and then in vitro is sort of the more artificial, you know, sort of cell culture system that we use to, you know, test specific hypotheses. So.
And Augustina, thank you so much for asking that question, because as we're collecting biosamples from our community and we're working in conjunction with Combined Brain, parents are asking, you know, how are these iPSCs going to be important for my child specifically?
Mm-hmm.
And so knowing that you can test these small molecules and later possibly other translational treatments.
Yes, yes. And thanks for bringing that up. I mean, I would love to keep in touch, and, you know, as we get further along in these studies, you know, maybe we can have some kind of a helpful collaboration where we can help families get more information about what treatments may or may not be helpful for their child. So
that would be great. Okay, well, with that, Dr. Knowles, thank you so much for joining us here today. It was a true pleasure, and we look forward to seeing you soon.
Likewise, pleasure to meet you all, and look forward to future collaboration and conversations.
Thank you. Bye.